Yeah. How many cells are there in our body? They say around 60 to 100 trillion cells. Just imagine a few of them, just a handful of them, declare autonomy. What would be the condition? It's going to be a chaos of highest order. It's not happening usually in healthy conditions, not happening. They work in collaboration with each other. So the cells which work together, the group of cells which work together for a common goal is called as tissue. Similar cells which work together for a common goal is called as tissue. So in that case, there are four types of tissue. The entire human body is made of four types of tissue. They are the epithelial tissue, connective tissue, muscular tissue, and the nervous tissue. Okay, even so, these tissues are put together in a different proportion to make the organ. Different organs work together to form the organ system. Different organ system work in hand to make a human body functional. So let's talk about the epithelial tissue today. Epithelium, the term epithelium means what? The definition, what definition you can give for epithelium? It's the one or more layers of cells which cover the outer surface of the body or the cavities inside the body, the luminal aspect of the hollow viscera. What do you mean by hollow viscera? The viscera which have got some cavities within it. For example, stomach, small intestine, urinary bladder. Okay, which has got some space inside that. It is called as hollow viscera. Or the luminal aspect of the blood vessel and the glands are called as epithelium. It can be just a layer or layers of cells which covering the outer surface of the complete body, or one or more layers covering the inner surface of the hollow viscera, or that layer which covering the inner aspect of the body cavities. What are the body cavities then? Peritoneal cavity of the abdomen, the pleural cavities of the thorax, pericardial cavity in which the heart located. All these are the body cavities. Okay. So one or more layers of cell. We are talking about all that only today. So based on the shape of the cells, there are three types of cells. Some are thin and flat cells. They are called squamous cells. Some cells are cuboidal in nature. That means it has got the more or less equal height and width. They look like a cube, hence it is called as cuboidal epithelium. Next type of cells are called columnar epithelium. Here the cells are tall column-like. That means their width is much lesser than the height. So these three types of cells are going to make the epithelium, that mainly. Then based on whether it's a single layer of cells make the epithelium or a multi-layer. So we divide the epithelium again into two categories. Simple epithelium means one single layer of epithelium lie over the basement membrane, or in stratified epithelium more than one layer of cells are located one over the other. Okay. So let's talk about the simple epithelium first. Simple epithelium, that means only a single layer of cells. That can be made of squamous cells, that can be made of cuboidal cells, or that can be made of columnar cells. So based on that, we call it as the simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium. The fourth variety is actually a single layer, but it look like a multi-layer. Hence it is called pseudo-stratified columnar epithelium. It's a single layer but appear like double layer. Hence it is called pseudo. Pseudo means false, you know it. So let's see where you have the simple squamous epithelium. Single layer of cell, simple. Squamous means the cells are thin and flat. Here you can see this is the simple squamous cells. The thin and tapering ends with less amount of cytoplasm in it, thin and flat nuclei. So this type of cells are called as the squamous cells. So this is the way it will be looking like when you see the side view. But on surface view, on a panoramic view, you can see the cells will be looking like a polygonal cells. The same cell is seen as a thin flat cells with a central flat nuclei, very little amount of cytoplasm. Okay, so they are naturally, metabolically very few amount of organelles and few amount of cytoplasm. So metabolically not much very active. The adjacent cells are connected with each other by a special type of cell junction called as zonula occludens, otherwise called tight junction. So that anything has to pass across the epithelium has to pass through the cells, not between the adjacent cells. Understood? It cannot pass with the gap between these two cells, but it has to pass across the cell. So there is the one which between the connection between the cell and the adjacent basement membrane is called as the hemidesmosome. There are different types of cell junction, zona occludens, zona desmosome, hemidesmosome and all. So where you can see this simple squamous epithelium, the typical classical example, we talk about the epithelium where you can see the squamous type is the alveolus of the lung. So this is the airway, the intrapulmonary bronchial tree which divides and divides ultimately enters into this small bubble-like or a grape-like structure called alveolus. This is a group of alveolus. Okay, alveolar sac. This is a single alveolus. The wall of the single alveolus is lined by thin squamous cells. So this is the one alveolus you are seeing here, and this is lined by a single layer of thin squamous cells. So it facilitates the exchange of gases between the alveolus and the capillary plexus lying over it. You can see here over the alveolus you can see a plexus of capillaries. Okay, the blood is taken into the lungs for the oxygenation. So the blood is having carbon dioxide in it. So this capillary blood has got carbon dioxide in it, whereas the alveolus is filled with the oxygen we inhale. Then now the exchange happens. The carbon dioxide will be taken into the alveolus and oxygen will be given to the blood in the capillaries. This is called gaseous exchange. In order to happen this in an efficient manner, the barrier between between the capillary and the alveolus need to be minimum, isn't it? So this thin squamous cell epithelium is the apt one here. So when you are taking a small section of the lung and do the histological processing, then stain it and keep under microscope having a thin section of 5 micron or something, then you will see this alveolus get compressed and collapse. So every thin line what you are seeing is the wall of the alveolus which is collapsed. It can be 1 or 2 adjacent alveoli will be fusing to make a bit more thicker blue lines. If you take a thin line, it will be made of a single layer of squamous cells. Done? Where else you can see the innermost layer of the blood vessel. This is the thickness of the blood vessel. It has got 3 layers from inner to outer. Okay, tunica intima, tunica media and tunica adventitia. In the tunica media we can see a layer of squamous cells. What we call it as the endothelium. In the blood vessel it is called as endothelium. If it is the inner layer of the heart, we call it as endocardium. Okay, If it is lining the peritoneal and the pleural cavity, we call it the mesothelium. According to the position, the name changes. Clear? So the function is to, since it's a thin, the separation between the two areas will be minimum, so it will be helpful in the exchange. Now simple cuboidal epithelium, just exactly like looking like a cubes here, isn't it? So that here the width and the height of the cell will be almost equal, so they appear like a cube. The nucleus will be circular and in the central position. This type of epithelium you can see in many parts of our body. The classical example we talk about is the thyroid follicle. Thyroid is an endocrine gland which is located in your neck. So here we can see that it is made of so many small, what do you call, something like a flask like or like alveolus. That's more like a bubble like structure. Okay, so this bubble is lined by cuboidal cells. Interesting time. So these individual cells are cuboidal epithelium. They produce the secretion and store in this alveolus, which will be released into bloodstream according to need. There is no duct for the gland. So this is called endocrine glands, ductless glands. Okay. Another area where you can see the cuboidal epithelium is the outer surface of the ovary. Outer surface of the ovary is called germinal epithelium, but it's a misnomer. It has nothing to do with the formation of the germ cells. Deeper to here happens the formation of the ova and all, but this is a simple covering called as the germinal epithelium made of cuboidal cells. I don't want to talk more detail about that now. Then where you can see again the proximal convoluted tubule, PCT, and then you can see the pigment cell layer of the retina and the choroid plexus, inner surface of the lens. All these are the area where you can see the cuboidal epithelia. You need to remember at least few of them. Simple columnar epithelium. Here you can see the cells, individual cells are tall column-like. The width of the cell is much lesser than the height. Around the height will be 3 times than the width. The cells are, the nuclei are basal, erect and oval. Basal, erect and oval. See, under light microscope, on the low power, you won't be able to see the cell membrane. How you make out what type of cell a particular cell is that, see in the shape of the nucleus. If you see a thin line like, you are going to assume it that it's squamous cell. If you just see a rounded dot, you can take it as a cuboidal. If you see oval bigger nucleus, you are going to take it as the columnar cells. Done? So here this kind of columnar cells, usually they are absorptive or secretive in function. You can see this type of epithelium in the stomach wall and the large intestine wall. Now there are some modification for the columnar epithelium. When we talk about the columnar epithelium, some cells, the columnar cells, some region show small modification on the apical part. The apical part of the columnar epithelium shows some modification. So they are called as the microvilli. It can be thread-like structure called as the cilia, or it can be smaller, the longer microvilli called as the stereocilia. So these are all modification for the functional purposes. Okay, let's talk about the microvilli. The tall columnar cell showing microvilli on its apical portion. Actually, what is this microvilli? They are nothing but the finger-like processes of the cell membrane. Cell membrane is throwing into folds. cell membrane in the apical region thrown into folds. So what would be the benefit? The surface area of the individual cell is increased. That means better, wider area for the absorption. So naturally this kind of the microvilli lined columnar cells can be seen in the small intestine. Okay, sometimes it can be in the form of uniformly distributed. In the case of small intestine, you can see these microvilli are uniformly arranged. It is called as striated border. But in the case of the gallbladder, if you see a small piece of gallbladder under microscope, you can see here and there clumps of microvilli are seen. This is called brush border microvilli. So what is the core of this microvilli? Cytoplasm of the cell. Okay. So they are the one mechanism to increase the surface area for the better absorption. See here under light microscope, you cannot see anything like a finger-like process here, but you can see one line here? Can you see one pink line here? This pink line denotes the microvilli. Okay. So this is the basal, rect, oval nucleus. This is the supranuclear portion of the cell, and this is the apical portion that bear the microvilli. Done. Let's talk about the stereocilia. It's nothing but the longer and branched microvilli, where you can see this kind of epithelium. So this kind of epithelium we can see in the epididymis, a small comma-shaped body on the posterior border of the testis is called as the epididymis. It's a storehouse for the sperm. So the sperm and the secretion from the testis will be taken into the epididymis and stored over the here the cells, the stereocilia in the epididymis will absorb the extra secretion reached from the testis. Okay, it's also seen in the receptor cells of the internal ear. They are longer than the microvilli and they are branched as long as something 5 to 10 micrometer in length. The next is the cilia, having cilia. Cilia are small hair-like processes. Okay, if you cut it, you can see it is made of one pair of microtubule in the center surrounded by 9 pairs of microtubule. So they are motile in function. It has got a tip, it has got a shaft, it has got a basal body. So this basal body is attached or continuous with the cytoskeleton of the cell membrane, just deeper to cell membrane. So they are motile. Okay. So what would be the function? Transport. They are there in your trachea. So if you happens to inhale any foreign particle like a dust or carbon particle, all these foreign particles, dust, everything will be deposited or trapped in the mucosa lining the trachea. So this mucus will be taken towards the pharynx by the beating of the cilia. Then you spit it out. So it helps in the transport of things. Another area where you can see is the fallopian tube. In the fallopian tube, it is lined by columnar cells lining having the cilia. So the ovum released from the ovum released from the ovary will be taken into the fallopian tube, and the beating of the cilia in the fallopian tube, the cells of the fallopian tube, helps the journey of the ovum towards the uterus. So it's the beating of the cilia is compared to like a wind blowing in a field. It start from one end and it moved to one direction so that it moved like a wave. First the distal part will be moving, then come the middle portion, then goes the proximal, that way, so that it takes the released ovum from distal part of the fallopian tube towards the proximal part, towards the uterus. It could be seen the medullary and auditory tube, ependymal canal or the central canal of the ependymal lining the central canal of the spinal cord, olfactory cells, vestibular or cochlear cells. All these areas it facilitates the transport. Learn, remember few of them at least. Okay. Goblets situated among the tall columnar cells. We can have some area under microscope which look like an empty space. Actually they are not empty space, that's occupied by a cell called goblet cell. Goblet means wine glass. Goblet means wine glass. So this is a unicellular gland which producing the mucus. So the apical area is filled with a drop of mucus that pushes the nucleus towards the basal area. When the tissue is taken and undergone histological procedure, this drop of mucus is lost. So under light microscope using HND stain, you can see this cell that it look like an empty space. Here, goblet cell. It's a unicellular gland. That means a cell acts as a gland. Okay, so it produces the mucus which uniformly spread over the mucous membrane, over the cell surfaces into which the cilia are projecting, and it acts a trap for the foreign material to get embedded into it. So we have seen the epithelium of 4 types. The simple epithelium, where the apical part doesn't have any kind of modification. Then we have seen the columnar cells having microvilli. Then we have seen the columnar cell having stereo cilia. Then we have seen the cells having the cilia on it. So 4 types of columnar cells. Now let's go for the pseudo-stratified cells. Pseudo means false. Stratification means more than one layer. That means it gives a false impression of stratification, but in fact it has got a one layer of cell. That means all the cells are reaching the basement membrane. Then how the false appearance happening? Because some cells are smaller, some cells are bigger, some cells are smaller, some cells are bigger, and the nuclei are located at the wider part of the cells. That means the nuclei are located at different levels. So it gives a false appearance under light microscope. I told you, under light microscope you cannot see the cell membrane. What you will be seeing only the nuclei. So if you just imagine you are seeing just nuclei in this picture, you definitely going to think that it's a multi-layer, because you are seeing many layers of cell 1, 2, 1, 2. So it gives a false appearance of stratification because the cells are of different size and the nuclei are arranged at different levels. That give a false impression of stratification. These cells are usually columnar, so we call it as a pseudo-stratified columnar epithelium. The classical example is the trachea, where the apical part of the columnar cell bears cilia. So trachea is an example for ciliated pseudo-stratified columnar epithelium. Okay. Pseudo-stratified non-ciliated columnar epithelium, you can see the stereocilia. The example is the epididymis. Clear? stratified epithelium, that means more than one layer. It can be of cuboidal, it can be of squamous, it can be of columnar. Okay, so based on what the top layers is made of, if it is a top layer is made of squamous cells, we call it as the stratified squamous. If the top layer is made of cuboidal cells, we call it as the stratified cuboidal. If the top layer is made of columnar cell, we call it as the columnar, stratified columnar cells, epithelium. Stratified columnar epithelium, the basal cell, the one or two or more layer, which can be the basal layer can be either cuboidal or polyhedral. But the top cells definitely going to be the columnar cells. Okay, this type of epithelium you can see in the larger ducts of pancreas, of salivary gland, and it also seen some parts of the male urethra. Stratified cuboidal epithelium, again the lower layer can be of cuboidal cells or columnar cell, but indeed the top layer is made of cuboidal cells. Okay, terminal ducts of the sweat or sebaceous glands are the one which is usually we consider as the stratified cuboidal epithelium. Stratified squamous epithelium. Stratified squamous epithelium is a multi-layer, the typical classical multi-layer, about 6 to 8 layers of cells you can see here. The basal layers are always columnar cell. On top of it, few layers of polyhedral cells. Polyhedral cells means the cells which are having many corners of varied shape and size. But the superficial layer is squamous cell. 2 to 3 layers of squamous cell. Since it is called as stratified squamous epithelium. Okay. So this is an example here. This you are seeing is a epithelium of the esophagus. You can see the basal cells are columnar. Middle cells you are seeing the rounded nucleus of the cuboidal cells, and the top layers are thin and flat squamous cells. Now we can divide the stratified squamous epithelium into two varieties based on whether the presence or absence of a protein called keratin. Okay, so it is divided into stratified squamous keratinized epithelium and stratified squamous non-keratinized epithelium. This is the keratinized, this is the non-keratinized epithelium. So let's talk about that now. Stratified squamous keratinized epithelium you can see on the dry surface. The typical example is the epidermis of the skin. You know, the skin has got two layers, outer epidermis and the lower dermis. Epidermis is the stratified squamous epithelium. The topmost layer is called stratum corneum. It's made of thin, flat, dead cells having no nucleus or having no any other cell organelles, but it is completely filled with a protein called keratin. Okay, so the cells in the basal layer multiply and it is pushed toward the surface. Takes around 6 to 8 weeks for a cell to migrate from the basal layer to the top level. As they reach the superficial layer, they are almost dead and they become thin and flat and completely filled with the keratin. So this keratin helps to prevent the dehydration. So it's a protective coating. Where you can see this, the epidermis of the skin, that means where the dry area located. Now stratified squamous non-keratinized epithelium. Non-keratinized epithelium. Okay. So here you can see there is no keratin coating. So here we'll be seeing the cells are nucleated and live cells. Unlike the non-keratinized epithelium, in non-keratinized stratified squamous epithelium, superficial cells are live. Okay. Where you can see this kind of epithelium you will be seeing in the case of oral cavity, pharynx, esophagus, vocal cord and vagina. Non-keratinized epithelium. The wet surface will be always wet. The superficial cells are live and no keratin over it. Transitional epithelium. It is somewhat similar to stratified squamous epithelium, but here the superficial cells, the top cells are not thin, flat squamous cells, but it is made of the big cells, big umbrella shaped cells. Okay, so here this type of epithelium is seen in the urinary system. It can be there in the minor and the major calyces, pelvis of the ureter, the urinary bladder, the proximal urethra are lined by this type of epithelium. Hence this epithelium otherwise called urothelium. Okay, what is the peculiarity of this type of epithelium? It can be stretched without tearing. In the urinary bladder, when the bladder is empty, you can see the epithelium will be of 6 to 8 cell thick. As the bladder gets filled, the epithelium becomes thinner. The superficial cells become thinner, but they never get detached from the adjacent cells. So without any wear and tear, it can be stretched. Okay. So when the urine is full, the epithelium becomes thinner, but when the bladder is empty, it assumes a thicker, the thickness of the epithelium will be more. Hence it is called transitional epithelium. Transitional, that can be, the epithelium can be stretched out to become thinner, but when the bladder is empty, they assume a epithelium which is of 6 to 8 thick epithelium. Okay, and here one more thing we have to remember here is that the cell membrane of this top cell has got a certain glycoprotein in it that prevent the seepage of the urine stored in the urinary bladder will not seep into the bladder wall and subjacent tissue, causing the harm for the organ. So this pilferage is prevented by a special protein that is called as plaque. So two things you have to remember is that it can be stretched without separating from each other. At the same time, it prevents the seepage of the fluid into the bladder wall. So this otherwise called urothelium. So here you're just going to see this much only the epithelium. The basal cells are columnar cell, and then you are seeing few layers of polygonal cells having rounded nucleus. The superficial cells are umbrella shaped cells. Some cells possess more than one nucleus, binucleated. So this is the urothelium or transitional epithelium. Next let's see. See here, this is a stratified squamous epithelium. There's a difference between the stratified squamous epithelium and transitional epithelium. I will tell you. In both areas, the basal cells are columnar cell. The cells are showing the basal erect oval nuclei. Then you are seeing a few layers of polygonal cells having rounded nucleus. But in the stratified squamous epithelium, you can see the top cells are having thin flat nuclei. The cells are squamous. So have got the thin flat nuclei. This black, sorry. So you are seeing the topmost cells showing the thin flat nuclei of the squamous cells. Okay. So this is the simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium. This is stratified squamous, stratified cuboidal, and stratified columnar. Sorry, the transitional epithelium, and this is the pseudo stratified columnar epithelium. So what they do for us? Protection. Yeah, you have seen in the case of skin, stratified squamous keratinized epithelium is a big protector for the internal environment from the external environment. Your body is completely covered by skin. So it's a very effective barrier of your deeper structure. Okay, from the chemical, physical injuries. Absorption from the lining cells, the lining columnar cells of the small intestine or the intestine, they helps in the absorption. Secretory function. The small intestine has got the glands, intestinal glands, or any glands. It can be the producing the enzyme, it can be producing the mucus, it can be producing the any hormones, so secretion. So actually the glands are the modified epithelium. Sensory, on the roof of your nasal cavity, the posterior most part of the roof, there is a small area where you have a few modified sensory cells, neurons. They are called olfactory mucosa. They are actually modified neurons, nerve cells. So they help in the sensation of olfaction. Exchange between the different areas. For example, in the case of the capillaries in the Bowman's capsule of the kidney and the epithelium of the filtrate. So here the barrier means it's a very effective barrier between two environments. For example, the brain blood barrier. Okay. So these are the functions. Thank you so much. Please give me the feedback.